Okay, happy Monday and welcome back to FirstThought Focus. For those of you who are new to FirstThought, we are a life sciences focused expert network with a mission to bring subject matter expertise back into the expert network industry. FirstThought Focus is our virtual conference series that aims to showcase the breadth and depth of FirstThought's experts, while also covering a wide range of topics that are relevant and important to the life sciences industry. We recently wrapped up a 4-part series on CAR T and cell therapies. You can find the replays for those at our website, firstthought.io/focus. And in the weeks and months to come, we will be covering many other topics, including oncolytic vaccines, more on the human gut microbiome, and also some medical conference recaps throughout the summer. I also wanted to just mention, we do want to hear from you. So if you have a topic you'd like us to feature, please reach out. We'd love to hear from you. And finally, just as a quick reminder, before we get started, we will be taking questions per usual from the audience at the end of today's discussion. Thank you to those who've already submitted some via email. That was great. If you would like to ask a question, you can submit them to the email address hello@firstthought.io. Again, that's hello@firstthought.io. And with that, I'd like to turn things over to our moderator and veteran science journalist, Neil Canavan. Go ahead, Neil.
Well, welcome. My name is Neil Canavan, and this is the first of 2 FirstThought webcasts that are going to focus on the human microbiome and its impact on health. Specific to today's talk, the microbiome's influence on the immune system when it comes to cancer. So I came to know something about this topic in writing my book. I wrote a book called A Cure Within. This was published in 2017 by Cold Spring Harbor Laboratory Press, where in the closing chapter is all about the nexus of the microbiome and cancer treatment, specifically cancer immunotherapy. If you'd like a free signed copy of this book, just mail your snail mail to Morgen, and I'll mail that thing out tomorrow. Now, you don't have to take my word for it that this is a pretty good book, because the person holding the book in photograph is none other than my guest today. And that is Dr. Jennifer Wargo. Dr. Wargo is a professor in 2 divisions of MD Anderson Cancer Center, the first being in the Department of Genomic Medicine within the Division of Cancer Medicine, and the second is as a professor in the Department of Surgical Oncology. Dr. Wargo, Jen, welcome.
Neil, thank you very much. And thanks to FirstThought for inviting me today to share my insights on the gut microbiome and health and cancer. It's a great honor.
All right, now, Ralph, really quick, I just want to represent here.
Hey, all right.
Hey, got my jacket on. So I want to set the table for our listeners today by referencing some facts in the Pulitzer Prize winning graphic novel, Microbiomes for Dummies. First, there are roughly 100 trillion, with a T, trillion microbes in us, outnumbering our own cells 10 to 1 and weighing in at about 5 pounds. Ooh. Of those trillions of bugs, roughly 10,000 distinct microbial species are represented in the human ecosystem, and every human is host to a unique collection of more than 1,000 of those species. As for functionality, while the human genome includes roughly 22,000 genes that we express, the microbiome has more than 8 million unique protein coding sequences. This means there are 360 times more active bacterial genes than human genes in our body. So where are these bugs? For those of you with small children, you may think they're all mostly found on toddlers. This is not exactly true. The majority of the bugs are in our gut, and most of those in the large intestine. As for what these bugs are doing, they're helping us digest food. They produce vitamins that we vitally need, and they help somehow regulate our immune system. Jen, I'm going to ask you to explain this mechanistically in a few minutes, but I'd like to start out with first, how did you become aware of this importance of the microbiome as it relates to cancer? How did it come on your radar?
Yeah, no, that's a great question. And so, with regard to my background, so before coming to MD Anderson, I was at Harvard University and the Massachusetts General Hospital, and phenomenal place where I studied response and resistance to different forms of cancer treatment. And when we were doing this, it's a phenomenal collaborative network there. And we were looking at resistance to treatment with targeted therapy, molecularly targeted therapy. And we actually worked with a group from MIT and the Broad to use this 3D culture system to interrogate the role of different cells in mediating response. And what was interesting is we found that there was a certain cell line that was able to confer resistance to treatment with chemotherapy for pancreatic cancer and colorectal cancer. And we were super excited about it. And then on routine testing of the cell line in the lab, it was actually contaminated with mycoplasma, which for anyone who is in cell culture, it's pretty devastating. It's a common contaminant and can really wreak havoc on experimental results. But Ravid Straussman, who was the lead on these studies, is really brilliant because he said, well, wait a second, what if these bacteria that are in the cell line are actually contributing to the therapeutic resistance? And so he tested it, showed that if he eradicated it, no longer conferred rescue. When he actually infected another cell line with mycoplasma, it could then rescue cancer cells from treatment with chemo. And sequenced it and found it was mycoplasma, and then took it back to a mouse model and showed that if you co-target the intratumoral microbes with antibiotics along with treating with chemotherapy, you could actually cure all the mice of their cancer. And we looked in patient samples and found very similar findings. We actually found in patients with pancreatic cancer, you could actually identify bacteria within the tumors of patients with pancreatic cancer in over 75% of them. And we actually found that the bacteria were actually breaking down chemotherapy into its inactive form. And we published those findings together in Science a few years ago. And so that's how I got started on studying the microbiome. Was actually, we kind of backed into it, if you will, and looked at the tumor microbiome. But then I had seen very elegant work that was done by Tom Gajewski's group on the gut microbiome and its role in shaping immune responses and cancer immunotherapy responses, presented at a meeting at the Society for the Immunotherapy of Cancer. And after the talk, we were happily studying the tumor microbiome. After the talk, saw this phenomenal talk that was done in mice, and got up to the mic and asked if they had studied in patients, and they hadn't, and saw a unique opportunity to do that. And so we started to study the role of the gut microbiome as well as the tumor microbiome in cancer.
Okay, you've mentioned a few names that I'm going to follow up on in a few papers. So I'm pretty sure I was in that audience for that talk. So the first I see of your publishing history is in 2016. It was a paper, Monitoring Immune Responses in the Tumor Microenvironment. And this was something you looked at as the microbiome. Is the work that you put on that paper, did it then become clear to you that I want to work in this, and this is going to be a major focus of my research?
Definitely. I mean, I think first studying the tumor microbiome, then hearing about, the gut microbiome, and we launched our studies, I think in 2013 or 2014, and very early saw a clear signal that depending on what the bugs look like in the gut of patients with cancer, they either responded or didn't respond to treatment with cancer immunotherapy. And furthermore, that things that could change the microbiome, like antibiotics or different probiotics, could actually change how they respond to cancer treatment. And so saw this as a real, kind of next new hot area to study and to help patients, right? And so really, I think this is foundational, not only tumor microbiome, but how the gut microbiome can influence our immune system, responses to cancer immunotherapy and other forms of cancer treatment, and saw this kind of as a new real pillar in cancer therapy.
So this idea that's new, and this occurred repeatedly in my book, Cure Within, there was a lot of pushback from the cancer immunotherapy people. Advisors were telling them this is stupid, companies were telling them this is stupid. Did colleagues push back on your sudden interest and this thrust into this field?
Well, I think we ourselves really wanted to be critical about the data early on. And, and again, in the tumor microbiome, if you think about it, finding that's such a critical finding, and you have to make sure it's right. And so we spent years and years, Ravid did proper controls for contamination, made sure this wasn't just, a fluke, and that these bugs were being introduced in the, in the context of tissue processing, et cetera, but no, found that these are indeed there, and worked on other ways of looking at this. And, but yes, there was skepticism at the outset. And, I'll go back to a comment that you made, so in our bodies, hundreds of trillions of microbes outnumber our normal cells. If you actually sample the genomic content within an individual or an organism, only 1% is human, but 99% is microbial. It's crazy, right?
Wow.
And so what's really changed things is that next-gen sequencing not only has allowed us to look more closely at tumors, in the host genome, but now at the microbiome. So next-gen sequencing approaches have really illuminated the impact of these microbes. And so instead of focusing on that 1%, should we now be shifting our focus onto that 99% that are inherently modifiable?
So I want to provide the capstone to the evolution of this science and the international focus it now has. As I said, I'm pretty sure we were in the same audience at SITC for Tom's talk, and he was talking about his mouse models. And then Laurence Fogel, Gustave Roussy, shortly thereafter, she published some findings, presented her findings, retrospective findings on microbiome and responders to certain types of chemotherapies. And then your talk, I heard, and then that's how we met. And then the three of you published back-to-back papers in Science in 2018. And that really blew the lid off. I mean, after that, the focus was without doubt. Now here, this is from this one of your papers, the Science paper I just referred to. Now, as the audience can see, this is all really simple. So just for fun, Jen, what the hell are we looking at, and what did you conclude from it?
Right. And so you can see, and I had to go back to my microbiology textbooks from medical school, but if you can see all these different names of bacteria here, either by 16S sequencing, which is one type of sequencing, versus metagenomic sequencing, which is a different. And basically, you see the blue here is responders, red is non-responders, and you can see that there are some bugs that are enriched in responders and others which are enriched in non-responders. And so, the question is, can we take this information and then derive novel therapeutics where we can actually target microbes to enhance immunity and responses to cancer immunotherapy? Now, that's really exciting. And, of course, now a whole industry has been formed appropriately to do just that, but there's nuances, and it's because if you think about it, when you look at all the studies that have been published, it's not the same bug that's popping up in every study, right, as being associated with response. And why is that? And there's probably a couple different reasons. Certainly, I think that it's the function of the bugs that matters a lot more, and then there's a lot of variables that go into individuals. Humans aren't mice, right? And we have different diets. I always say that the diet in Houston, Texas is very different than the diet in Paris, France. I love Paris and I love Laurence. And, can these diets actually impact responses to cancer treatment and the gut microbes? And totally the answer is yes. But appropriately now there's a whole industry focusing on this, not only on engineering consortia of microbes that could be used, but prebiotic strategies, postbiotic strategies, various forms of manipulation and dietary interventions, which could actually move the needle, not only in patients with cancer, but on overall immunity in healthy individuals, ultimately. Wouldn't that be nice? And really contribute to better overall precision health.
I want to go back to the paper for just a moment. So I have two questions related to that. The first is pretty simple. What was your sample size? And the second is, did you break it down by, quote, super responders versus partial?
Right. Now, great questions. And, our sample size in that original paper was around 50-ish. I think 54 was the total. We actually now have a follow-up study where we studied a couple hundred patients and we see the same signals. And we also focus on dietary patterns in patients. And we see that if patients eat a high fiber diet, they're more likely to respond to cancer immunotherapy. And we have parallel studies in preclinical models in those studies. Those aren't yet published, but I think, galvanize really this notion that gut microbes are important in immunity and in cancer immunotherapy response, and that we should be taking every advantage to try and manipulate these, but do it cautiously and with data.
Right. Now, as I said, these papers really touched up a floodgate of microbiome research. There were microbiome-only sessions at conferences. There are now, I think, 1 or 2 microbiome-only conferences. But I want to go back to the first I mentioned of this topic and how mechanistically this makes any sense. Granted, I'm not an immunologist, but the fact that these trillions of bugs are actually educating our immune system kind of blew my mind. I'm like, how does that make any sense? So this is a graphic I want to put up now. This is from a review paper that Dr. Wargo and several others published in Science in March of this year. Now first, Jen, if you could pick out an example or 2 from this and walk us through the mechanism of how bugs are affecting us. How does it make sense?
Yeah, exactly. No, this is a great paper that Greg Poor and Rob Knight, Laurence Zitvogel and I and others and Jeff Hasty put together, and really kind of to get out there to the scientific community. This was published in Science a few weeks ago, and really kind of gives a whole high level but broad and comprehensive view of how microbes impact us, not only at the level of the gut, but also at the level of the tumor, and potentially even circulating microbes or microbial signatures can actually impact our health, and definitely cancer patients, right? And so I think, there's a couple of examples here, and Laurence Zitvogel has done really foundational work, as has Tom Gajewski and others, in showing how gut microbes can impact our immune system. And if you think about a lot of the microbes that exist in our bodies exist in the gut, okay? And all along that long alimentary tract, which is, many meters squared of surface area, on one side you have, these trillions of microbes, and just on the other side, you have this dense network of infiltrating immune cells. And there's this constant and dynamic interaction, kind of a to and fro. And so you can have different aspects where these microbes are actually educating immune cells, not only T cells, but also dendritic cells and other types of immune cells, which can be educated in the gut and then even home back to lymph nodes or tumor or elsewhere in the body and actually mediate responses. Another way is by metabolites. And if you think about, all the metabolites that exist in our blood, a large fraction of those are actually derived from microbes. And, these can actually have distant effects. And so can metabolites that are being secreted by microbes actually have an impact on the immune system and other processes, distantly? And the answer is totally yes. If you think about the gut-brain axis, that's certainly one way as well. There's that gut microbes have been implicated in things such as autism and other neurologic conditions. And so we're still uncovering all of the ways through which gut microbes can actually impact not only immunity, but other physiologic and pathologic processes. But there's certainly a tremendous amount, thank goodness, for all the work that's been done by people worldwide, because we're learning so much, and it's definitely here to stay, if you will. The microbiome is here to stay, and we're going to be able to derive not only strategies to profile gut and other microbes in overall precision health and in cancer, et cetera, but also ways to manipulate it.
Well, I'm going to make it really simplistic because that's how I do things. So would part of this education be, okay, here comes the microbe, here comes the dendritic cell or the T cell, sample, 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 sample. Okay, I know you're fine. And you go off and do it work. But if that's missing, all of a sudden, wait a minute. Now I'm going to go attack that thing. Is that part of it?
Yeah, exactly. And so if you think there's tolerance to these microbes, right, because they have to live in, coexist with us. They actually help us. They help us digest food. They produce metabolites. They shape our immune system. And so there's tolerance to some of these gut microbes. And if you break that tolerance, you can actually have things like autoimmunity, and so what's interesting is if you think about over the past several decades, as we've, really embraced more widespread use of antimicrobials to reduce infectious disease, as you see the levels of infectious diseases go down, you see other conditions go up, like autoimmunity, right? And are we actually creating another problem by, use of, say, broad spectrum antibiotics every time someone has a cold or, the sniffles or something, and granted, we need antibiotics when they're needed, but can we use a more targeted approach? And are we shooting ourselves in the foot sometimes by potentially over-treating and and really impacting microbes within our gut or at other sites.
Yeah, I mean, you're referring to like the hygiene hypothesis that we're just being too clean. And I want to just toss out a couple of examples, one from 2019 and one from, I think, a week ago. 2019, this guy published in Nature, he was a UK researcher, and he sampled, I think, 70 or 80 newborns. And he looked at those born by C-section and those born vaginally. And there was a marked difference in their microbiome. So A, does that make sense to you? And he didn't follow them. He hasn't followed them up to see if they're less healthy or more healthy. That result just right there makes perfect sense to you?
Yeah, I think if you think just passage through the vaginal canal during birth, certainly there's exposure to microbes that you don't get when you're in the sterile environment of an operating room and delivered, through an incision into the uterus. And it makes complete sense that you would have some differences in the microbiome at an early age. And could this actually impact overall health ultimately? I think breastfeeding too, if you think about breastfeeding versus bottle feeding, and different aspects of early development, it's worth thinking about early on about the impact of these microbes, not only from method of delivery to, choice for, breast versus bottle feeding versus other foods. And then, how about, and I hate to take, open a can of worms, but how about, the food that we're eating? And, are there antibiotics that have been used in the process of raising, either chickens, cows, what have you, or, preservatives, et cetera. Like this, it's, it does kind of blow the roof off, not only cancer treatment, but all kinds of, public health issues with regard to the microbiome.
The other one I wanted to mention that's really quite recent. By the way, Jen Wargo is a mom and she has a dog. So this goes, and happy Mother's Day belatedly. So they mentioned the dog, and this research came out, and this guy looked at infants that had a dog in the household versus not. And he followed them over time. And these kids with dogs, grew up with dogs, have far less childhood allergies. I mean, so yeah, just be kind of filthy, play with your dog.
Yeah.
So now I want to move on to, now I think we've got a lot of basic science here. Let's
well, I would say too, Neil, Rob Knight, real leader in the microbiome space, wrote a book called Eat Dirt, and I have a copy of it myself, and it's like published a few years ago, but I think, I was actually going to reach out to Rob again and see if he wanted to co-author a book, and kind of bring it up to date and with more data and implications. But I think, there were a couple of papers, or there was a paper published in the New England Journal a few years ago about different methods of farming, one where they had where they basically, worked a lot with the animals and had them in their houses, and another where it was very industrialized, and the families that had that worked more alongside their animals actually had a lower incidence of asthma, and it was a beautiful paper published in the New England Journal, so I mean, it's, it goes to show. Now, granted, I think there are nuances here, and we shouldn't just all go and eat a lot of dirt, but, is there an advantage or disadvantage to having environments be more sterile?
Okay, I would suggest a possible title for the book is Let Your Dog Lick You and Don't Think About
It. Yes, that's right.
Okay, now I want to move towards what's actionable, but there is, there's one other basic science discovery you touched on already, and this is fairly recent, and it was very surprising to me when I heard it. And that's how microbes are actually being found physically in tumors themselves and directly influencing tumor progression. So I want to put up another graphic. This again is from Dr. Wargo's review paper. So Jen, this is sort of the universe that you sampled. Does this have real-time implications for how cancer patients are initially evaluated?
Yeah, I think, again, this is something we started to study, probably about a decade ago with Rabin Straussman, who's a true leader in this space. And he published a paper in science in May of 2020 showing that pan-cancer, he could identify bacteria within tumors, across multiple cancer types. And this includes brain cancer, GBM, right? And so does this have implications? Absolutely. In addition to this, Rob Knight published a paper in March of 2020 with Greg Poor, where they profiled the TCGA, or the Cancer Genome Atlas, and basically mined that data using very specialized techniques for decontamination, et cetera, and identified, again, microbial signatures across all cancer types, not only in the tumors, but also in the blood of patients with cancer. And so does this have here and now implications? Totally it does, but I think we need to understand it better. One, we need to understand what microbes are in different cancer types and how that relates to, not only how the cancers develop themselves, but how they respond to different forms of therapy. Also circulating microbial signatures. There are actually companies that are now focused on liquid biopsy approaches, where they actually in Rob Knight's paper showed that you could discriminate between cancer and non-cancer by using this liquid biopsy approach focused on the microbial signatures. And so could we have a liquid biopsy approach focused on that along with other variables, and then, again, I think ultimately, can we target these microbes to improve response to cancer treatment and hopefully prevent cancer altogether? And, there's a precedent, right? HPV, you look at HPV. So we're actually now mining not only bacterial signatures within patients with cancer, but also viral and other signatures to see, what's there. Doing mechanistic studies to see, validating it, is it real or is this noise? And then doing mechanistic studies to say, okay, is this a driver event? Did this actually contribute to the development of the cancer, or is it a passenger, and how is it impacting therapy response? And then how can we target it, right? Can we develop novel therapeutics, and that's with regard to the tumor microbiome.
All right, well, I'm going to get to the actual treatment approaches by various companies in a few minutes. I just wanted to mention to our listeners, Dr. Wargo also did a research paper, a review paper in Nature in 2020. This is AI finds microbial signatures in tumors and blood across cancer types. So if you want to do a deeper dive, have at it.
Yeah, exactly. And Neil, just to clarify, that was the News and Views coverage of Rob Knight's paper.
Oh,
so
okay.
It digests it well for the, less AI, savvy individuals. So it makes it a bit more comprehensible. They went through some very elegant processes, which we boiled down and put into a little News and Views piece.
Okay. So actually one more science question. This is very general, and I've asked you this before, and last time you go like, eh. So here's the question again. You see a person with a cancer, the disease of cancer, their microbiome is all screwed up according to your definition of screwed up. Did the dysbiosis, this imbalance, occur before the cancer, or did the cancer cause the dysbiosis?
Great question. And I think, we and Laurence and others are really studying this. And I think it's a little bit of both. I think, certainly Bob Schreiber's elegant work on, cancer immunoediting, could a dysbiosis in the gut or other microbial niches contribute to the development of cancer because your immune system is altered, right? And because you're less able to have that cancer immunosurveillance, are you more likely that if a cancer cell arises, are you more likely for it to set up shop and grow and potentially metastasize? Early work would suggest potentially yes, but I think there's still a lot more work that needs to be done. And I know Laurence and we and others are really focusing on this. And if so, how? How does this happen? And, what are the mechanisms through which this happens? So then the other question is, how does cancer impact the microbiome? And so within the context of the gut microbiome, just by implanting, say in preclinical models, just by putting a cancer into a mouse, totally changes their microbiome. It's crazy, right? So there is this to and fro, and, it's really wild, right? And so it's both basically, but cancer can impact the microbiome. The microbiome definitely can impact cancer. And then it's finding that sweet spot. How can we actually get to a point within, I'll pick the gut microbiome, at which we have, kind of a perfect harmony, if you will, that, we can not only, prevent cancer by adequate immunosurveillance, have good vaccine responses to SARS-CoV-2 vaccine, et cetera. Can we have that perfect immune function based on our gut microbiome composition and inherent genetics, that we can then hopefully prevent cancer altogether and be overall more healthy. But we have to be feeding it the right things too.
So I'm going to start touching on some procedures and potential business plans. But first, I'd like to point out that as you alluded to earlier, 2 entirely or 5 entirely healthy people all over the planet who don't have cancer, Houston, Paris, Cairo, have entirely different microbiomes, and yet they're fine. Mm-hmm. So from a business angle, that's going to be a bit of a puzzle. Now, the first treatment, and this is the most obvious, though least palatable, is FMT, or fecal, or let's just say it the way kids do it, they're poop transplants. Now, she published some results in Science on this quite recently, actually in February. Fecal microbiota transplant promotes response in immunotherapy refractory melanoma patients. So just briefly, what did you find? And how did you correct it with poop?
Yeah, well, this is very elegant work that was done in Israel by Erez Baruch and Gal Markel, Ben Borsi, and others. And I had actually seen the work presented at AACR annual meeting a few years ago, after, of course, we had published our papers, and they had gone the next step and run this trial where they basically took patients with metastatic melanoma who had progressed on multiple lines of prior therapy, either immunotherapy, even targeted therapy, and then wiped out their inherent microbiome, transplanted in a totally new microbiome from a patient who had had a complete response or had metastatic melanoma, got immunotherapy, and all their tumors went away, right? Those patients donated poop samples. They depleted the inherent microbiome, gave them a poop transplant, and then retreated them with immunotherapy. And 30% of those patients responded, which is pretty phenomenal for that patient population where they're highly refractory. And, it was really impressive because not only did they do this clinical study where they looked at responses by imaging, et cetera, by CAT scans, but they also actually did biopsies of the tumors themselves and also of the gut, the colon, because they actually delivered the FMT, or in part through colonoscopy. And what they found is that treatment with that poop transplant was actually, you got more immune cells in the tumor and you also got more immune cells in the gut, but it depended on who the donor was, at least in that study. And then there was a second study that was published out of the University of Pittsburgh, back to back, and showed again in 15 patients that if you use poop transplant, you could actually reverse resistance to immunotherapy. And 6 out of the 15 patients had clinical benefit. So pretty compelling, right? So this could actually work. But I think it's tough, and especially in the days of the pandemic, poop transplants have risks, and certainly they've been used a lot for C. diff and other diseases. But there have been cases where patients have gotten multi-drug resistant organisms. Some patients have died from those, with reports in the New England Journal. And so there are a lot of efforts, and, they, worldwide, at the FDA and other places to really ensure safety of these approaches as much as possible. And they're doing a great job. But, can we ultimately get beyond fecal transplants? I think someday we will, but I think in the short term, fecal transplants are potentially a great way to treat this, and definitely very worthy of studying this.
I did attend a lecture a while back, and this one physician was addressing the issue of compatibility. And he suggested that you use the spouse, because after all, quote, they've been taking each other's crap for years. Yeah. Now, so let's talk about some direct approaches. I'm going to mention some approaches. I'm not going to put company names to it because we don't want to do that here, but I'll just talk about the approach. So there's several companies that use their monoclonal single species supplementation. These are almost all of these approaches I'm going to mention are oral supplement. Yes, no, thumbs up, sure, What do you think?
Yeah, I mean, I think a lot of definitely worth, and need to be studied. So, these are simpler to manufacture, which is great because, if you think about fecal transplant, getting that into a pill, and getting it to patients is really hard, right? People have done a great job at it, but it's still very difficult. So can you identify that single bug that would be associated with improved responses to treatment? And, how could it be a single bug? Well, it could be because, you could have a single bug that could stimulate the immune system against a type of cancer. And sometimes there's this molecular mimicry where there's,
Right.
it looks very similar to, the cancer itself. And through an approach like that, you could nail it, right? And so I think they're tractable. I think it might be cancer specific. We're not sure. It's not going to be necessarily a one size fits all unless you have that perfect bug that, is going to stimulate dendritic cells in the exact right way, that's then going to turn on the immune system to fight cancer, specifically without having a lot of autoimmunity. But, it's I don't know, after looking at all these studies where you look and it's a different bug in each cohort that's associated with response, are we going to find that one single bug? I think it would be great if we did. I think it's a little bit nuanced, and these first generation approaches might work for specific cancer types. I think we might need to shift and focus more holistically on the overall complement of bugs in the gut. And then it's how do these single bugs mix with the other bugs that are in there, is an important question. If you're taking a pill with 10 to the 11 CFUs or colony forming units of a specific bug, is that going to disrupt the inherent microbiome? Will these bugs play well in the microbial sandbox with the inherent microbes, and I can tell you, for the studies in which fecal transplant's been used, the couple of studies that were published, they actually wiped out the inherent microbiome and then transplanted in an entirely new one. Now, with these monoclonal microbial approaches, would you want to do that? I candidly would worry a little bit that if you wipe out all the bugs in someone's gut and then transplant in one, you're going to have a very low diversity microbiome, and you might not do well, and you might even harm, so I think it's really nuanced. And we published a paper in Lancet Oncology in 2019, Jen McQuaid and I, on different approaches to modulate gut microbes, and all the, I say it's like all the things that keep me up at night, including my 4-year-old, but the many different aspects of microbiome manipulation that keep me awake at night.
Well, I mean, the other thing about whether you put in more bugs or one bug, it goes back to my question about dysbiosis. Is like if your system is imbalanced and it's steady, and you throw it off of that new balance, won't it just return to where it was with the same dysbiosis? So this we don't know. So to cover your bets, perhaps a consortium, a cocktail of bugs. Other companies are doing that. That make a little more sense, or again, let's see the data.
Yeah, I mean, I like the approach of the consortia in the short term too. I think, because if you think about it, when we looked at our patients, it was patients who had a higher diversity, who were more likely to respond to treatment. And it wasn't necessarily that the diversity itself was important, but I think it's because you had a more diverse microbiome. It could kind of fill holes in the functional repertoire, of other microbes. So if you were missing certain ones, they could sub in for another one. So I like the idea of consortia, but how many is enough, and how scalable is it? It's hard to grow these bugs in, big vats, and have a stable consortia that could be used, and how do you manufacture that and administer that? And so people are working on that. I think that has a lot of promise. And I think the consortia approach is very tractable.
Okay, now a different approach, I know of at least one company doing this, is they're going to bypass the use of bugs altogether because they feel it's not a pharmaceutical business plan. So the way they looked at it is, we're going to figure out what the bugs are actually doing. What metabolites are they shooting out that are affecting you? And we're just going to use that, and that will be the drug, potentially small molecule drug. Your reaction to that approach?
Yeah, no, I think, I always say function over phylogeny. It's not the names of the bugs that matters. It's what they're doing. Now, I think can we get to that in the next few years? I hope so. That would be great, because I think it is, from a functional standpoint, where we need to be. But will it require, depending on the extent of dysbiosis or how profoundly dysbiotic an individual is, say, with cancer, is it enough to give a small molecule drug that mimics the impact of these gut microbes, or will they actually need, more of a drastic FMT or consortia, or, the right monoclonal microbial?
Okay, so two more approaches now I want to mention, and this would be, as opposed to using a grenade, using a laser. Phage. So as opposed to antibiotics, which are general, phage are extremely specific. Could you use a phage to remove the offender?
Yeah, exactly. And I think that is another great approach. Now, if you think about, is it the presence of a good microbiome, the absence of a bad one, or a little bit of both? It's probably a little bit of both. But, with regard to certain aspects, there are, for toxicity example, Laurence and I recently did a study together where we actually looked at patients on combined immune checkpoint blockade targeting CTLA-4 and PD-1, studied a cohort that had toxicity to therapy, compared them to those who didn't have toxicity, and identified a single microbial taxa that was associated with the toxicity. And could you actually use, say, a phage to knock that bug out? And that's super tractable, right? And, validated in a second cohort, I think, really interesting and definitely a potentially very tractable strategy. Interestingly, in one of the studies of C. diff years ago, they actually studied the supernatant and showed the same effect as transferring in the bugs, for treatment of C. diff, which suggests that could it be the phage or other aspects that are associated with this. And that's, something that we're now, because I think a lot of time and effort was spent early on bacteria and different taxa that are associated with response, non-response toxicity. But now I think we're looking beyond that, using metagenomic sequencing and looking at phage, viruses, you name it, and so I think we will get a lot more insight into that. But yeah, I, use bullets, not bombs. I think, the phage approach is very tractable.
Okay, now something it's everywhere in science. It's everywhere. Everywhere. I'm going to put it on my sandwich later, and that's CRISPR. How about that?
Yeah, definitely. I love it. Because talk about, it's not the names of the bugs that matters. It's what they're doing. Can we use CRISPR technology to change function, right? And even take someone's inherent microbiome, ex vivo, manipulate it, put it back in. You hopefully avoid some of the safety concerns that you have with FMT and other strategies. How scalable is that? I don't know, but I think it's a great, up and coming approach in microbiome modulation, maybe here and now. And I haven't seen many studies published on it, but I think it has tremendous potential.
Okay, one other.
And I'm putting it on my sandwich later too.
All right, so one other company business plan, this several companies, I'm sure, it's roughly 23andMe for your bugs.
Yes.
Is this information actionable?
Yeah, it's tricky, right? I know I always say like 23andMe for your poop, right? Can you actually do, some microbiome profiling that would really be actionable? Ultimately, yes, I think so. I think it's been tricky early on, and there are companies that have tried to do this, and the data's not there. We can't overstate or make claims, and so I think it's really tricky right now to be able to provide that kind of information, and it's not truly actionable right now. That being said, keep your eye on this, because there's things like smart toilet is being developed, right, where you actually sit down, you put your finger on or thumb on the handle. It basically gets information on who you are. You go to the bathroom, it analyzes your microbiome, sends data back to your smartphone, And hey, could we have an actionable strategy based on that? Wouldn't that be cool, huh? You know, and then how about volatile organic compounds? You know, so there's VOCs, right? Which are, you know, in our breath. Could we do a breath biopsy someday, you know, and find out, are we dysbiotic? How good is our microbiome? You know, and ultimately, could we actually, you know, on my smartwatch, you know, could I either breathe into something or, you know, go to the bathroom and all of a sudden I'll get an alert on my watch, you know, Jen, it's time to eat some more kale. You know, it's in our lifetime, Neil. You know, I think
No,
it's going to be there.
No, my objection is to the kale.
Yeah.
So I just want to toss out a couple more questions, then we're going to go to the questions that have been coming in, which is, let's talk about a couple things you may not want to do. Antibiotics in cancer patients.
Yeah, I think it's buyer beware, if you will. You know, I think broad use, you know, use of broad spectrum antibiotics without clear indications should really be critically thought about. You know, so Laurence Vogel in her paper in Science showed that if patients with cancer, lung cancer, took antibiotics just before or just after starting treatment with immune checkpoint blockade, they had worse survival, right? And pretty dramatic, but you know, there was some skepticism because those patients are sicker, that's why they were on antibiotics. And so, but now multiple studies have been published. David Pinato had a great paper in JAMA Oncology in 2019, very striking differences. If they took prior, if they were treated with antibiotics just before starting on immune checkpoint blockade for cancer, they had definitely had worse outcomes. And now multiple studies have been published. We're actually running a study at MD Anderson where we are asking the question, can a single dose of perioperative antibiotics used in the context of surgery actually deleteriously impact the microbiome? You know, and so we're running a clinical trial, you know, and are we over-treating, you know, and it's conceivable that we are.
Okay, and I'm going to add one more and then we're going to get questions. And this I've asked you a while back and you're like, no, please don't quote me, I'm going to get in trouble. Probiotics.
Yeah, and it's a hot topic, right? And so I think, you know, with the data emerging on the gut microbiome and its impact on immunity and overall health, you know, people are really excited about changing their microbiome. And, you know, there's pills, right? You know, you can go out to CVS and there's probiotics, and, you know, they're, you know, on the label, it says it improves health, right? You know, but does it improve health for cancer patients on immunotherapy? And so we asked that same question. We found that in a relatively small cohort, we saw differences, but there were, you know, didn't meet that 0.05, you know what I mean? Like, so we didn't really necessarily see statistically significant differences in outcomes between those who reported taking versus not taking probiotics, but it inspired us to study a little bit more, because we did see a hint that patients who took over-the-counter probiotics might actually be doing a little bit worse, right? So we took it to a mouse model and treated, basically took germ-free mice, gave them the best microbiome we could, then gave them either sterile water versus one of two probiotics we bought off the shelf, and found that if they received the probiotics, they did worse when they were treated with cancer immune checkpoint blockade. Whereas, wow. You know, so you know, now it's not all probiotics are bad. We don't know which ones are good, but they need to be studied better, right? And I think in patients with cancer who are going on to cancer immunotherapy treatment, think hard about, you know, what you're putting in your body, right? And, you know, if run it by your doctor, and if it's not studied, you know, you should probably avoid it, and think about better ways to favorably impact your immune system by eating fiber, you know, by whole fruits and grains, you know, again, consult with your doctor and your team. But I think, you know, there's a lot of things that we're learning about that have an impact, you know, that may or may not be intuitive.
Okay, so that's my prepared questions. Morgen is now going to jump into the questions that have been coming from the audience.
Thank you both. That was really great. We have a lot of questions, so I'll see which ones we can get through in the time that we have left. But Jen, to start, I wanted to ask, are the effects of the gut microbiome on cancer and immunotherapies due to the production of short chain fatty acids?
Yeah, it's a great question. And one that we thought, our hypothesis going into this was slam dunk. It's totally on short chain fatty acids. We saw hints of this, but it's not the only thing that's mediating this. So I think though they play a major role, it's not the be all and end all. And I think it's more complicated than that. Which is exciting because there's more than one avenue to treat it, but it's also, a little bit complicated. It's not going to be a quick fix and an easy find, but yes. So can they play a role? Yes, they can. It's not the primary mechanism through which, at least in our studies.
Got it. Thank you. Also, we had a listener asked today, can you speculate about why consumption of a high fiber diet would increase responsiveness to immunotherapy?
Yeah, no, it's great. And it's like it's interesting because we studied this in a cohort of patients and found that if they ate, if they consumed a high fiber diet, more than 20 grams a day, or sufficient fiber, we should say, that they were much more likely to respond to treatment. And worked with Giorgio Trinchieri at the NCI, who had run studies in mice and showed that if mice receive a low fiber diet, they fail to respond to treatment with immune checkpoint blockade for cancer. And if they receive a high fiber diet, they do. And it's because they can activate their T cells. Now, what are the direct mechanisms through which this fiber is impacting T cell activation pathways within the tumor? We're still teasing it apart, and looking at the different contributions of different cellular subsets at the level of the gut and the tumor. But, I can tell you across the board, what we've seen in patient cohorts and now in preclinical models, it's definitely true. And, can we mimic that with a prebiotic type of a strategy? I think would be the ultimate goal. I think, because dietary interventions they work, but they're hard, right? Compliance is not always easy, so we actually have a chef at MD Anderson and a kitchen where they prepare foods for patients. But, compliance can be an issue, right? And could we come up with either a pill prebiotic that someone could take, or new industry? Could you actually have a food industry that's focused on, these engineered diets that would actually improve immunity, et cetera? So I think there's opportunities there as well.
Awesome, thanks. Next question. Based on your February publication using super responding donors for FMT treatment in non-responders with melanoma, I know we touched upon this a little earlier. Is this a strategy that could be adopted for other cancers? What are your thoughts here for usage more broadly for non-responding patients with late-stage terminal cancers? Is FMT something that should be looked at more often in these cases?
Yeah, it's a great question. And I think, we certainly are looking at this, and there are other investigators around the world looking at this, because not only do we see this in patients with melanoma, but others have seen this in patients with other cancer types. And so we and others are exploring the use of fecal transplant or FMT from complete responder donors, or even healthy donors in some cases, into patients with other cancer types, including pancreatic cancer, colorectal cancer, multiple different cancer types. And, the trials are early, so we don't have data back yet, but the suspicion, or my hunch and hypothesis, would that be absolutely we can impact. Because if you think about it, most patients with advanced cancer, even, after having been treated with chemo, other approaches, their gut microbiome is pretty dysbiotic, and oftentimes we'll see that there's an element. It's not just the microbiome that's making cancer grow or progress or, become resistant to treatment, but it's a big part of it, at least with treatment with immunotherapy.
Thanks, Jen. Next question. This sounds like zoology on steroids, or akin to figuring out our genome. How many different microbes have been ID'd thus far in the body, and how is AI helping to solve for this?
Yeah, great question. Wow. And it's like you're only, I always say we started off with bacteria because the reference databases are better with bacteria, right? And you by doing next-gen sequencing and metagenomic sequencing and looking at the different microbes that are in tumors, gut, circulation, or microbial signatures, then you can get some clarity into who's there and what they're doing, right? Now, again, you're only as good as your reference database. And so most of the bugs that have been well characterized are bacteria, but now viruses and fungi are also emerging, and yeah, I mean, there's still a lot to learn. There's still a ton of work to be done, but, total transformation of healthcare, I think, in overall precision health through the microbiome.
Just got another question come in. To what extent do we think that each tumor type has a distinctive population of bacteria?
Mm-hmm. Yeah, and probably some of the best work on this has been done by Ravid Strassman and by Rob Knight. I think, again, early days, we need to do deeper dives in different tumor types and actually very carefully prospectively collect these samples so that we minimize contamination. But these will be tumor type specific, or at least there will be components of this, that are tumor type specific. And if you're seeing a single bug across multiple tumors, it may be real, but it may be contamination. But, if you think about some of these viral signatures too, like, these viruses are, it's raining down viruses on us every day, and they're integrating even into our host genomes. And could these be contributing to cancer and into cancer treatment? Totally, yes. But I think we need a lot more studies on this, and happy to have many people studying this worldwide, and hopefully will inspire more to study it, because I think it's not only is it a hot area of study, but it's going to have huge implications and help our overall health, and also help patients with cancer.
Great, thank you. Hopefully we have time just for a couple more. Next question, Jen, is the manipulation of microbiome applicable only for immunotherapy or for conventional chemo as well?
Yeah, great question. I think so. Laurent Zitvogel actually did a lot of work on patients with chemotherapy and showed differences in the microbiome. There may be, it depends on the chemo, no blanket statements apply here. I think you need to study the type of cancer, the type of treatment, how is that treatment being used? Is it after multiple other lines have been used, or frontline, So I think it's very likely that the microbiome will have an impact not only on chemotherapy response, but maybe even, how that chemotherapy is metabolized and, within an individual. So yeah, absolutely. But I think, again, a lot more data, is needed to be able to say, for any given patient, what impact their microbiome has on their treatment.
If I could jump in, we just have 1 or 2 minutes left, and I do want to address the zoology on steroids question. Jen et al. at MD Anderson are working on this furiously, obviously. Jen, can you tell us about Prime TR?
Oh, yes. Yeah. So Prime TR, catchy name. It's the Program for Innovative Microbiome and Translational Research at MD Anderson. And we basically, the institution saw the value of studying the microbiome and has invested in it big time. And basically, we have a program that's now focused across the institution, and then we're working with others worldwide to profile, to understand the mechanism, and to really manipulate microbial niches, not only at the gut, but other areas, in order to further cancer treatment, and ultimately, hopefully, have angles in cancer prevention.
Fantastic.
So thank you, Neil, for the plug. It's I love this program. It's great. It's like,
absolutely. I do anything for you. You know that.
Thanks, man.
So, hey, listeners, that's a wrap. Jen did mention very early in our webcast that microbiome has also been associated with CNS disorders like autism. Actually, our next microbiome session will focus on that. And our speaker will be a man named Sarkis Mazmanian. I know that sounds like an evil overlord from a planet somewhere, but Sarkis is a wonderful man and a terrific speaker, and I encourage you to tune in.
Great. Thanks, everyone. Stay safe.
Take care.